Anatomy of flowering plants.
One. Tissues in plants. Meristematic tissues, actively dividing cells found in growth regions. Apical meristem, located at root and shoot tips, responsible for primary growth. Intercalary meristem, found at internodes or base of leaves, responsible for the elongation of internodes. Lateral meristem, located along the sides of stems and roots, responsible for secondary growth, vascular cambium, cork cambium. Permanent tissues, derived from meristematic tissues but have lost the ability to divide. Simple tissues, parenchyma, thin-walled, living cells, involved in photosynthesis, storage, and secretion. Parenchyma provides mechanical support. Cells are elongated with unevenly thickened cell walls. Sclerenchyma, dead cells with thick lignified walls, provide structural support. Complex tissues. Xylem conducts water and minerals, consists of tracheids, vessels, xylem fibers, and xylem parenchyma. Phloem transports organic nutrients, composed of sieve tubes, companion cells, phloem fibers, and phloem parenchyma.
One. Function. The primary function of xylem is to transport water and dissolved minerals from the roots to the leaves and other parts of the plant. It also provides mechanical support to the plant due to its thick, lignified cell walls. 2. Structure. Tracheids. These are elongated, tube-like cells with thick walls and tapering ends. They are dead at maturity and have pits in their walls that allow water to move between adjacent tracheids. Tracheids are the main water-conducting cells in gymnosperms, like pine trees. Vessels. Vessels are also tube-like structures, but are made up of a series of vessel elements stacked end to end. These cells have larger diameters than tracheids, and their end walls are perforated or entirely dissolved, forming long, continuous tubes. Vessels are more efficient at water transport and are predominantly found in angiosperms, flowering plants. Xylem parenchyma. These are living cells with thin cell walls that store food and help in the lateral conduction of water. They can also help in wound healing and regeneration. Xylem fibers. These are elongated, thick-walled cells that provide additional mechanical support to the plant. They are dead at maturity and have a supportive role rather than a conductive one. 3 types of xylem. Primary xylem, formed during primary growth from the procambium. It is found in the young parts of the plant. Secondary xylem, formed during secondary growth from the vascular cambium. It constitutes the wood in woody plants. Four. Direction of transport. Xylem transport is unidirectional, from the roots to the leaves. Phloem. Detailed description and structure. 1. Function. The primary function of phloem is to transport organic nutrients, especially the products of photosynthesis, mainly sucrose, from the leaves to other parts of the plant where they are needed or stored. 2. Structure. Sieve tube elements. These are elongated, living cells that are aligned end to end to form sieve tubes. Their end walls, known as sieve plates, have pores that allow the flow of phloem sap. Unlike other living cells, sieve tube elements lack a nucleus and other organelles to allow for efficient nutrient transport. Companion cells. These are specialized parenchyma cells that are closely associated with sieve tube elements. They contain a nucleus and various organelles, providing metabolic support to the sieve tube elements. They also help in loading and unloading sugars into and out of the sieve tubes. Phloem parenchyma. These living cells are involved in the storage of nutrients and help in the lateral conduction of food. Phloem fibers. Also known as bast fibers, these are elongated, sclerenchymatous cells that provide mechanical support. Unlike xylem fibers, phloem fibers are usually living at maturity in primary phloem but dead in secondary phloem. 3 types of phloem. Primary phloem, formed during primary growth from the procambium and is found in young plant parts. Secondary phloem, formed during secondary growth from the vascular cambium and is part of the bark in woody plants. 4. Direction of transport. Phloem transport is bidirectional, meaning that nutrients can be transported both upwards and downwards depending on where they are needed in the plant. Key differences between xylem and phloem. Xylem transports water and minerals, consists mostly of dead cells, and has unidirectional flow. Phloem transports organic nutrients, consists mostly of living cells, and has bidirectional flow. The tissue system. Epidermal tissue system, outermost covering of plants. Epidermis, single layer of cells covered with a waxy cuticle to reduce water loss. Stomata, pores for gas exchange, guarded by guard cells. Trichomes, hair-like structures on stems and leaves, reducing water loss and offering protection. Root hairs, extensions of root epidermal cells, increasing surface area for absorption. Ground tissue system consists of parenchyma, parenchyma, and sclerenchyma tissues. Cortex, located between epidermis and vascular bundles, mainly parenchyma. Endodermis, innermost layer of cortex, regulating the flow of materials into the vascular system. Pericycle, a layer just inside the endodermis, from which lateral roots arise. Vascular tissue system, conductive tissues, xylem and phloem. Vascular bundles, arrangements of xylem and phloem. Open type, with cambium, capable of secondary growth, example, dicots. Closed type, without cambium, no secondary growth, example, monocots. Anatomy of dicot and monocot plants. Dicot stem. Presence of a well-defined epidermis, cortex, vascular bundles arranged in a ring. Open vascular bundles with cambium leading to secondary growth. Monocot stem. Scattered vascular bundles, no secondary growth due to closed vascular bundles. Presence of ground tissue instead of distinct cortex and pith. Dicot root. Presence of a vascular bundle in a radial arrangement. Xylem and phloem arranged in an alternating manner, forming a star-shaped structure in the center. Presence of pericycle and endodermis. Monocot root. Similar to dicot root but with more xylem and phloem bundles. Presence of pith in the center and larger, well-defined endodermis. Dicot leaf venation. Dicot leaves exhibit reticulate venation, where veins form a network or web-like pattern across the leaf. This means the veins branch out and interconnect, creating a net-like structure. Stomata. Stomata are more concentrated on the lower epidermis, underside, of the leaf, which helps reduce water loss while still allowing gas exchange. The mesophyll in dicot leaves is differentiated into two distinct layers, the palisade parenchyma and the spongy parenchyma. The palisade layer, located beneath the upper epidermis, contains closely packed cells rich in chloroplasts for photosynthesis. The spongy parenchyma, found below the palisade layer, has loosely arranged cells with air spaces for gas exchange. Veins. The veins in dicot leaves vary in size and form a complex network, helping to support the leaf and distribute nutrients and water efficiently. Bulliform cells. Dicot leaves lack bulliform cells, which are specialized cells that help in leaf folding. Common examples of dicot leaves include those of rose, mango, and hibiscus. Monocote leaf venation. Monocote leaves have parallel venation, where the veins run parallel to each other from the base to the tip of the leaf. This pattern is simple and linear. Stomata. Stomata are equally distributed on both the upper and lower surfaces of the leaf, which facilitates uniform gas exchange. Mesophyll. The mesophyll in monocote leaves is not differentiated into palisade and spongy layers. The cells are more uniformly distributed throughout the mesophyll region. Veins. The veins in monocote leaves are generally of similar size and run parallel to each other, which helps in the efficient transport of water and nutrients along the length of the leaf. Bulliform cells. Monocote leaves contain bulliform cells, which are large, bubble-shaped cells on the upper epidermis that help in the folding and unfolding of leaves to conserve water during stress conditions like drought. Common examples of monocote leaves include those of grass, madge, and bamboo. Secondary growth in dicots vascular cambium. Forms secondary xylem, wood, inward and secondary phloem outward. Cork cambium. Produces the periderm, protective tissue, replacing the epidermis in older stems and roots. Annual rings, growth rings formed due to variations in activity of the cambium during different seasons. Heartwood and sapwood. Heartwood is the non-functional, dark central part of the wood, while sapwood is the lighter, functional outer part.